Dr. 𝗟𝗏𝗙𝗪 Shuja, Salim Habib University. I would request every presenter to be very precise on time because we have a lot of presentation in this session. You have 10 minutes.
My slide is not. That's CML presentation. CML presentation. Bismillahir-Rahmanir-Rahim. Start by the name of Almighty Allah, the most beneficent and merciful. I want to congratulate the whole team of LIFSCICON conference for putting up a great show, and thanks for providing the stage so I can share my verbal talk. My topic that is on investigating the role of novel cardenolides in chronic myeloid leukemia, targeting the RAS signaling pathway. Okay, let me tell you about the brief introduction of CML. CML is the chronic myeloid leukemia. It is the very important issue worldwide, and it is and cancer is the most leading cause of death globally. CML, if I want to, you can say that categorize the CML, it is the myeloproliferative disorder, which is due to the equal translocation between the chromosome number 9 and chromosome number 22, which causing a fusion of a new and very deadliest chromosome that is called the Philadelphia chromosome, and this is the main culprit behind the pathophysiology of the CML. If I talk about the epidemiology, leukemias composed about 15% of the adult leukemias. Yes, cardenolides. Cardenolides, these are the you can say these are the parent molecules which contain carbon-23 and lactone in their ring. These are the steroids molecules, but the anti-cancer activity is immense in a sense that they have a great potential to inhibit the cancer cells, which I have checked, that is on the K562. This is the in vitro work. So they have the excellent potential to inhibit not only the proliferative manner, but also provides an apoptotic pathway through which the cancer cells may evade and protect themselves, and they are not killed. So it is in a sense that they are anti-proliferative, apoptotic, and obviously they are also providing a mechanistic approach, so which I later on describe. Okay. Cardenolides are, if I tell you that it is traditionally used in the congestive heart failure patients, because their mechanism of action is that they work on the sodium potassium ATPase pump, and this provides a sustained mechanism for the continuity and functionality of the heart. But again, these are the very toxic substances. Okay. During the screening, I have like around checked maybe more than 50 compounds, but the most promising activity which I have found is on the cardenolides. This is the overall mechanism of cancer cells. What cancer cells does, that they usually causing the genetic instability along with the cellular changes, due to which, and again they also, like in a continued manner in the you can say that the cell cycle, so they never gets the resting phase, and as a result of which they are continuously producing and proliferating. Events in CML, yes. There are so many things which we need to understand while working on the cancer cell line, or either directly cancers. Because cancers is a very, you can say cancer very complex disorder, and disease because it works in a very mysterious way and in a very unprecedented way because they cause the evasion of the apoptosis and metastasis, local invasion, releasing of the growth factors which provide them a space so they can form the angiogenesis by releasing different factors including the VEGF, vascular endothelial growth factors, hypoxia-induced factor 1. So these are the factors that facilitate and providing the way and pave the way for making the blood vessels. Signaling pathway. There are so many signaling pathway, but in my case, 𝗬𝗏 pathway, that is RAS mitogen activated protein kinase mechanism. RAS is a extracellular receptor when the ligands attach to the receptors, it causing the transformation changes, and this due to the happening of the GTPase pump, and it causes the activation of the downstream molecule, and a result of which following things happen, which include proliferation, like cell division, differentiation, and maturation. Yes, significance. Significance that now in the recent era, the resistance and the retaliation is the main issue regarding the anti-cancer drugs because they are continuously changing their molecular mechanism, their dynamics are so tricky to be tackled. So it creates a need for us to investigate or discover the newer pharmacological moieties or compounds which have the excellent potential to inhibit the cancers. Methodology. Screening of the compounds, then after screening, I have selected particular compounds on the basis of IC50 value, and this IC50 value is the inhibitory concentration 50. For instance, at least 50% and above 50% value, if you don't get this value, your compound not having the enough potential for curtailing the overall mechanistic pathways of the cancer cells. Then I have checked the toxicity on the normal cell line, that is the BJ cells, human fibroblast, and after this, MTT. MTT is a very important and worldwide accepted calorimetric test, and this MTT is basically used to assessing the cell viability and proliferative capacity of the cancer cells. If I talk about the overall phenomena of MTT, MTT means that these are the tetrazolium salts, and the cells, those are viable, may convert the tetrazolium salt into a blue formazine, and the presence and the amount or the magnitude of this formazine may, you can say it is a confounding factor, and it provides that the cells are viable because the amount of formazine present in the 96-valpate will decide how much the active or viable cell count are there. Yes, again, RNA, gene expression, RNA, I must say, is a very laborious process, and I want to tell you in a very snappy way that from RNA isolation using the trizole method, and then cDNA synthesis using a thermoscientific kit method, and after that, that is the RT reverse transcriptase method. So I have checked the role of different genes, which include the BAX and the main pathway gene, that is the HRAS, and caspase-3, because this is the apoptotic gene. Statistical analysis, I have applied the one-way ANOVA because I want to check the mean difference between and within the group, so the value which is less than 0.05 may consider as a significant value. These are the compounds like quercetin, rutin, cardenolides A and cardenolides B. These above mentioned that 24-hour and 48 hours. Okay, so these on the basis of 24-hour value, I can be more decisive that whether I want to take this compound further on and check the mechanistic and protein values. So the very miraculous results what I achieved in case of the cardinolites. You can see the 24-hour value, that is the 0.05 micromole. That is a very, very least concentration, and in this concentration, it may produce around you can say 52 or around 54% of the killing of the 𝗂𝗵𝗲𝗀𝗃 cells. In the 48-hour value, again, that is 0.25, and in case of cardinolites B, that is 2 micromole. Again, it is a very nano concentration, and if this concentration providing the immense results, so I would say that cardinolites may have a greater potential as a new therapeutic approach in future. But again, clinical research and further research should be conducted to get the final results.
Last minute.
These are the morphological patterns. These are the CML cells. And in the cardinolite A and cardinolites B, you can see that the overall architecture of the cells are so much disturbed that they were prone to a apoptotic pathway, and this disturbance in the overall architecture and cell morphology, it suggests that yes, it is that the cells are killed by the cardinolites. Gene expression, 𝗏𝗏 and caspase-3. Okay, the first is the control value, then the DMSO. First one cardinolite A, cardinolite B, and the last one is the standard drug, that is the doxorubicin. In case of 𝗏𝗏s, the cardinolites A increases the 𝗏𝗏's concentration, and this provides that this is you can say it is a pro apoptotic pathway, and after achieving and getting the values in this in favor of 𝗏𝗏s, I checked the caspase-3, and this caspase-3 was also increased. But in this scenario, cardinolite B provide promising results. Yes, HRAS. This is the pathway gene. The pathway gene is also diminished, so it causes
Excuse me, it's already 12 minutes, so you are requested to please conclude it.
Okay. Last but not least, I must say that cardinolites are the family that belongs to cardiac glycoside. They have a great potential, and yes, indeed, future research should be conducted to achieve the final results. And yes, I have checked, because I'm presenting the one part of my study, I have checked the protein inhibition as well, mitochondrial role, electron microscopy I have also done. So this is the one part of my work. So I want to conclude my presentation. Thank you so much. Any question? The house is open. Would you like to ask any question? Yes, sir.
So actually, the plan is we'll be asking questions after these sessions, right?
Thank you.
So next presentation is plant-based protein solutions for One Health by Ms. Aasafa Shahid from Izzaddin University.
Bismillahirrahmanirrahim. Assalamu alaikum. My name is Aasafa Shahid. I am currently serving as a lecturer in Izzaddin University, and I'm also pursuing my PhD in Department of Food Science and Technology from University of Karachi. Today, the topic of my presentation is the plant-based protein solutions for One Health, nutritional and sensory enhancement of the flatbread with deamidated rice protein. Okay, so this is the list of the contents here. I'm going to introduce, giving the introduction about the One Health, how it links, how that my topic links with the One Health. Then I will share the research objectives, including followed by the objectives. I will share the research methodology, then the findings, and conclusion and future prospects. So here you can see. My research supports the One Health concept by promoting the plant-based proteins as a sustainable, healthier alternatives to the animal proteins. We contribute to the SDG 3 by offering the nutritious food options that reduces the risk of chronic diseases, reducing the dependence on animal proteins. Also supports the SDG 13, 14, 15 by lowering the environmental footprints, conserving biodiversity, and minimizing the habitat disruption that can lead to the zoonotic diseases outbreaks. Additionally, using the eco-friendly methods like 𝗏𝗡s aligns with the SDG 6 by reducing the chemical pollution, thus protecting water systems and ecosystems. Overall, this research advances the sustainable food systems that are better for human health, animal welfare, and the environment, that core vision for the One Health. Here, how about the food sustainability? Yes, it connects the human, animal, and the environmental health, forming a core part of the One Health approach. The shift towards plant-based diets play a key role in preventing the chronic diseases while reducing the environmental impact of food systems. Additionally, food waste valorization supports a circular bioeconomy by transforming byproducts into a valuable resources, promoting a more sustainable future. According to the National Nutrition Survey in Pakistan, 2018, approx 40% of the children under 5 in Pakistan suffers from the stunting, a condition linked to chronic protein deficiency and poor dietary diversity. What is green processing? Green processing approaches emphasizes sustainability and safety in food innovation. Natural deep eutectic solvents offer a eco-friendly and non-toxic alternative for the protein modification, particularly deamidation. Unlike harsh chemicals, 𝗏𝗡s are the biodegradable and derived from the natural compounds. These can improve the functional properties of proteins such as the solubility, emulsification, and foaming, making them more suitable for the diverse food applications. 𝗏𝗡s here we produced using the glucose and citric acid, making it a safe and sustainable medium for the protein treatment. My research objectives include to valorize the rice-derived protein using the green processing, to evaluate the nutritional and sensory enhancement of whole wheat flatbreads, and to explore the alignment with One Health through the sustainable innovation. This is the research methodology. You can see here the rice-derived protein isolates. So what is the rice-derived? It's basically a glucose industry agricultural waste which is currently used as a livestock feed. We will valorize this agriculture waste, which is called the rice drag. It contains a 40 to 50% protein in it. We will do the modification. Once we isolate this after the alkali solubilization and by isoelectric precipitation, we were able to collect the rice drag protein isolates. But the tendency of these proteins are basically they are limited in the food applications because in a wide variety of pH they are insoluble. So we have to do the modification of proteins here. For this, we have several options like chemical. In chemical, we can do the acylation, phosphorylation. But here we will use the deamidation process. We can also opt for the physical methods like heat treatment, high pressure, ultrasonic sonication, UV radiation, etc. And sometimes the biological and enzymatic treatment can also be used. But since here I am using the green processing, the deamidation is opted. And I've told you earlier that the glucose and citric acid solution we are using here to modify the protein isolates. You can see the white powder. The crude is converted into the white powder, which is called the deamidated rice drag protein. We characterize this protein isolates. We check its structural changes, and we check the solubility. Oops. Okay. The second part contains we will incorporate these protein isolates into the whole wheat flour. We prepare the dough from it. We check the dough characterization by using the Farino graph, which will check the absorption of the water, its dough handling, and dough stability. After that, we prepared with this composite flour, we prepared the flatbreads using a roti maker, automatic roti maker to contain the consistent thickness. Then we check the characterization of these flatbreads using the tensile strength by using the UTM, and then the glycemic index by starch solubilization, and the sensory analysis, also the color analysis we checked. So here in the research findings, we checked the mixture of different NADs were prepared, and we were able to see that the negligible solubility was achieved below the 14 into 10 to the power minus 3 molar. So 14 is the good ratio, and below that, the solubility further increases. The deamidation, the solubility of rice-derived protein isolates in different concentration of glucose, citric acid-based NADs. And we can also see the deamidation enhance the solubility of proteins and relax their structure bonds, as the deamided rice proteins digestivity was improved. Further, we can see the structural changes. This is a SZABIST size exclusion chromatography. You can observe the elution time at 6 minutes. The number one is a citric acid, and the control. You can see at 6 minutes, the highest elution was observed, which we can say it's a high molecular weight. And in the last one, you can see the deamided rice protein. This and later shift to towards the 13 minutes. It means the structural relaxation of the structure has been done due to the deamidation. In the picture A, B, C, D, you can see A is the crude rice track, which we obtained from the industry as a raw material. And then we have done the defatted, removal of the fat. You can see the color changes, the rice protein isolates, and then the deamided rice protein through the deamidation. Once the flatbreads were prepared, the tensile strength was checked. You can see the less and less force was needed. You can see in the green highlighted area, the less and less force was needed to break these strips of the flatbread, because it means that the flatbread became very soft due to the solubilization. Compared to the control and RPI required very less force to tear the strip because it was very soft. And here you can see the 1, 3, and 5%. We prepared the flatbreads with different proportions of the rice RPI and deamidated. So here you can also see the glycemic index. Some proteins can inhibit the starch degradation enzyme that can extend and thus reduces the GI. For the GI, we have already taken the very good lecture by Dr. Tahira that shows the low GI, how low GI is important in our dietaries. For the color measurement, we use a colorimeter in DEAM. A is steric with the LAB values increase, which means that the samples are becoming redder, and you can see it's the control 1%, 3%, and 5%. This is the native rice protein isolates, and on the B section, you can see it's a deamidated rice protein isolates. In both, the C is a control. It means it has no supplementation of the rice protein. And the sensory analysis was done using their aroma, flavor, taste, and it was found that the 3% deamidated RPI flatbreads were the most liked by the most of the audience. Conclusion and future prospects says that yes, the incorporation of a deamidated rice protein into the flatbread significantly improved the nutritional quality, dough functionality, and sensory attributes towards its potential as a viable plant-based protein enhancer in staple foods. Here we use the flatbread as a staple food, and the use of rice track via green chemistry offers an eco-friendly approach to a protein enrichment, contributing to a waste reduction, clean processing. You can see I have not used any harsh chemical for the deamidation or the modification of the protein, and alignment with the One Health and SDG framework. Future research could explore the deamidated rice protein isolate incorporation into other cereal-based products, evaluate the long-term health impacts, and assess industrial-scale feasibility and consumer market option. One Health integration, yes, it links with the human health, the environmental health, and the agriculture sustainability. These are the references. One of the research about the deamidation has already been published in the International Journal of Cereal Chemistry. And I would like to acknowledge my project supervisor, Dr. Mohammad Abdul Haq, the chairperson and associate professor, Department of Food Science and Technology, University of Karachi. Thank you.
Thank you so much for a nice presentation. The next presenter is Mr. 𝗵𝗲 𝗣𝗿𝗀𝗀𝗀 from 𝗧𝗵𝗲 University, and his title of the presentation is in silico structure and functional SNP analysis of rabbit melanophilin.
Ladies and gentlemen, Assalamu alaikum. This is 𝗵𝗲 𝗣𝗲𝗀𝗀𝗀 from second year of Biosciences from 𝗧𝗵𝗲 University. And topic of my research presentation is in silico structural and functional single nucleotide polymorphism analysis of rabbit melanophilin protein. Before I move into my research, let me tell you about objectives of my research. First of all, we had identified and characterized the single nucleotide polymorphisms in a gene of interest known as MLPH gene. Basically, SNPs are also known as mutation, in which a single nucleotide is replaced instead of another, due to which a different amino acid is coded, which leads to the change in the functions, stability, and folding of a protein. Secondly, we had predicted impact of those SNPs, that due to that SNP, what is basically the impact on the protein chain of the melanophilin protein. But before, let me tell you about my research. What made us curious to do this research, to do this study? So that was this genetic mystery. As you may see, these are rabbits which are from a same parent. These are siblings of each other. But the coloration pattern of on their skin, on their fur, have a difference. You may see here also, like, as these all rabbits are from similar parent, but the coloration pattern on their fur is different. So what was the main reason behind this? Why? अगर मतलब उनके माँ बाप एक जैसे हैं, उनके ऊपर एक coloration pattern आ रहा है, जबकि बच्चों के अंदर different coloration pattern आ रहा है, ये क्या चीज़ है? So basically, who was the main agent behind this mystery? So after studying, we had identified that agent was melanin hormone. Melanin is depicted as biological agent responsible for creating diverse range of skin tones. As you may see, in this individual, there is a high intensity of melanin. In this individual, there is bit less, then more or less in this individual, and least intensity of melanin is in this individual. So, but why rabbits? Why not on other animals also? Like there are cats, birds, and there are different animals on which the skin fur pigmentation is observed. So why rabbits? So the reason behind this is, when we talk about about genomic index, so 88% we humans and rabbits are similar genetically. And when we talk about protein functions, so when we talk about protein similarity, so around 92 to 95%, the protein functions in us and rabbits is similar. Moreover, they are also a lower animal model, which makes our study feasible to study. Also, they have diverse coat color patterns, which give us easiness to study this. Also, they have high reproductive rates. Rabbits reproduce very rapidly, so which give us rapid generational studies. And also, there is a limited research done on the local Pakistani rabbit breeds, which gave us novel opportunity of study. So this is my research, and this is how we had used our tools and methodology. First of all, we went to GenBank. We had studied several genes responsible for this coloration pattern I have told you. So there were some of the genes which are responsible for the production of melanin hormone. Some of them were responsible in regulation of that melanin hormone. But our gene of interest was MLPH melanophilin gene, which was responsible for the distribution of that pigmentation pattern. So we had chose that gene. Then we went to NCBI, National Center of Biotechnological Information, in which we took sequence of that gene. Then we performed blast protein, in which we compared the sequence of similar gene in different breeds of rabbits. Then By comparing those all sequences of similar gene, we had predicted SNP single nucleotide polymorphisms, that there was a point where mutation was occurring, due to which there was a change in the pigmentation pattern in the different breeds of rabbits. Then we also predicted the stability of that protein by predicting its primary, secondary, and tertiary structure. Then we also predicted the Ramachandran plot to check the stability of our protein of interest. Then we also predicted binding sites in that protein, which will further help us in docking activity also. MLPH, our gene of interest. It is known as traffic controller of melanocytes. Mutation in this MLPH gene lead to the change in melanosome transports within the melanocyte cells, due to which different pigmentation patterns are observed on the different breeds of rabbits. Also, it regulates the melanin distribution by facilitating the movement of melanosomes within melanocytes. These all are the single nucleotide polymorphisms we had observed. Like you may see, D13E. D is an amino acid, also known as aspartic acid. When, first of all, it was on the 13th position of the protein chain, but E amino acid, also known as glutamic acid, replaced it, due to which SNP occur. These all are the tools. You may see µPro, Polyfin 2, Provian, iMutant 2.0, and these all are tools which predicted the impact of that SNP, that due to this SNP, what is basically the effect of that SNP. So µPro told us, due to this SNP, there is a decrease in stability of protein structure. This Polyfin 2 told us that there is a benign effect, means this effect does not cause cancer. Provian told us that there is a neutral impact due to this mutation on this protein structure. And iMutant 2.0 told us there is an increase in stability. Same goes here also for different mutations, also you may have observed. And we had also predicted this diagrammatically. As you may see, when there was a serine on there was a serine on the 48th position of protein chain, and when on the 48th position, cysteine replaced it. So this was a change. But ये सब करने का क्या फायदा है? So when we observed this phenotypically, so first, when there was D amino acid on the melanophyllin protein chain, so this was a coloration pattern observed on the rabbit. When E amino acid, glutamic acid, replaced it, so this was a coloration pattern observed on the rabbit. Isn't it fascinating? Same goes here also. Here also, when serine was replaced by cysteine, this goes here also. Same goes for here. This is how the distribution of melanin happens. This is our gene of interest. This gene works along with these all genes by forming network. If a single gene among these all get disturbed, so our gene of interest will also stop its working. This is business model. We can prepare dyes using melanophyllin protein. We can use this melanophyllin in textiles, in cosmetics, in skincare products, in gene analysis, and in humans. We can replace plastic surgery and treat an individual with melanophyllin protein. We can replace this, we can perform skin cancer treatments, scar treatments. We can use this in forensics. We can modify natural skin color of the individual using this melanophyllin protein. I would like you guys to watch short documentary of my project along with my colleagues, that how we had performed this working. We are excited to unveil a groundbreaking research project, in silico characterization and functional single nucleotide polymorphism analysis of rabbit melanophyllin protein.
Our journey began with a fascinating observation. We noticed rabbit breeds with various fur coloration. Intrigued by this genetic mystery, we decided to delve deeper.
This need to find out more sparked our curiosity and led us to explore the genetic underpinnings of fur pigmentation. Our study focuses on the melanophyllin, MLPH gene, a key player in melanosome transport within melanocytes.
Rabbits share significant genetic similarities with humans, making them an excellent model for studying genetic mutations.
Rabbits serve as a model organism for understanding this, provide insights into the genetic processes in humans.
Genetic polymorphism in the MLPH gene can dramatically influence the coat color pattern. By studying these polymorphisms, we can uncover the molecular mechanisms driving coat color diversity, which means we can now modify or decide the coat color pattern of any organism.
We harness the power of bioinformatics tool to unravel the single nucleotide polymorphism in rabbit melanophilin gene. Our toolkit SZABIST, polyfin 2, PhD SNP, 𝗵𝗄 iMutant 2.0.
Our objective was clear: to classify these SNPs based on their potential to reshape protein function, stability, and ultimately phenotype.
We uncover several pivotal mutations within the MLPA gene, each with the potential to influence fur coloration. These changes include glycine to valine, aspartic acid to glutamic acid, and threonine to alanine.
By using multiple computational tools, we predicted the profound effect of these mutations on the protein structure and function. It evaluates the genetic mechanism behind fur coloration pattern.
The potential application of our research extends far beyond academia. Melanophilin can be used for the preparation of dyes, food colorants, and other coloration activities, offering numerous industrial benefits.
Our findings also align with existing research, highlighting the role of genetic polymorphisms in creating coat color diversity in rabbits and other mammals. This study sets the stage for future research aimed at deciphering the molecular intricacies of coat color diversity.
Experimental validation of our prediction will be the next thrilling step of our research.
Together, we are a team of passionate researchers under the expert guidance of Dr. 𝗵𝗄 𝗂𝗏.
Our work ignites curiosity and inspires future studies, unraveling the complex relationship between phenotype and genotype.
These are some of the references we had used for our study. This is my team. Thank you so much.
Thank you very much for the very interesting presentation. Now we have next presentation by Ms. Sajja Zahra from SZABIST University regarding biosynthesis of polyhydroxybutyrate derived biofilm plastics from thermophilic bacteria strains. Ms. Sajja Zahra.
Assalamualaikum and good evening to all of you. Thank you for the introduction. So my research topic is biosynthesis of polyhydroxybutyrate-derived biofilm plastics from thermophilic bacterial strains. So we all know that conventional plastics are derived from petroleum, which are non-degradable. They accumulate in the environment and cause toxic harm to our ecosystem. The annual production of these non-degradable bioplastics worsens each year, which affects not only our environment, but our ecosystem. To overcome this problem, there was an alternative eco-friendly solution, which is sustainable, biodegradable, and biocompatible. That is PHB. So what is PHB? PHB is a type of a biopolymer that is naturally produced by microorganisms to store energy under nutrient-limited conditions. It has similar properties to synthetic plastics like polypropylene. And as I said before, it is biodegradable and biocompatible and contributes to various resources and applications that we can use in our agriculture or biopackaging. So since it requires nutrient deficit conditions, I thought of a way to use sugarcane bagasse as a carbon source from which the bacteria can obtain its nutrients from. Sugarcane bagasse is readily available. It supports our circular economy as well as it is cost effective. Coming to our objectives, we wanted to determine between 3 thermophilic strains to see the PHB production. That is 𝗵𝗏 𝗂𝗏10, 𝗬𝗵𝗲 𝗂𝗏 and 𝗬𝗏𝗏𝗀 𝗂𝗏27. Then evaluate sugarcane bagasse as a carbon source and then optimize the conditions using different factors and prepare and characterize our PHB biofilm plastics between the 3 strains. After that, we wanted to test the biodegradability under seawater and garden soil. So the isolates were acquired from Karachi University, and they were revived on nutrient agar at 60 degree for 24 hours. After that, gram staining was done on the 3 strains. Then screening of PHB was done to see which strain produces PHB. So again, it was revived on nutrient agar, and then it was incubated at 60 degree for 24 hours. Then it was stained by Sudan black beet dye, and then it was destained by ethanol for 30 seconds. The same procedure was done on the second medium that we wanted to use for our PHB synthesis, that is nitrogen deficit medium. So we did the same process on a different medium. Then the third screening was done, that is microscopic screening. For which we stained the slides, heat fixed them with, and then stained again with Sudan black beet dye, decolorized it with xylene, and then counter-stained with safranin. After that, we viewed the plates under 100 magnification, under microscope. So for the extraction of PHB between the 3 strains, first, sugarcane bagasse was used in the nitrogen deficit medium. After that, we inoculated our bacterial strains and then incubated for PHB production. Then we centrifuged the strains at 4,000 RPM for 40 minutes to collect the bacterial cells. After that, the supernatant was discarded and the pellet was saved. The pellet weight was then weighed, and then we put 0.9% saline and then mixed to wash the cells. After that, the tubes were then again centrifuged at 4,000 RPM, and the supernatant was discarded. After that, the pellet was then dried at 100 degrees C at hotter oven, and the pellet weight was recorded. After this process, 5 ml of bleach and 5 ml of chloroform was added and shaked for about 1 hour at the shaking incubator at 60 degrees C. After that, it was then again centrifuged, and then the bleach layer was discarded, and the chloroform layer was filtered out. After the filtration, it was dried on the plate again at 60 degree, and the weight of the PHB was calculated. After our extraction process, we use the statistical software, that is Minitab 18. On this, we used it made 3 designs, experimental designs, that is PBD, Plackett-Berman design. It was used to screen out the variables single-handedly, which was temperature, inoculum size, carbon source, pH, and then it generated 12 runs, which had different factors that were single-handedly screened. After the PBD, followed by the PBD, CCD was done, which generated 16 runs, and it was done in triplicates. It further saw the interaction between 2 factors. So PBD was single-handedly screening 1 factor, and this was seeing, CCD was seeing the interaction between 2 factors on our PHB yield. After CCD, RSM was done. After the result of CCD results, RSM calculated further temperatures to see out of these which temperature produces the most yield. And it generated 3 additional experiments which had variables temperature, pH, and inoculum size. After this, one run order was selected which produced the most yield, and it was carried out further. Then our characterization was done through SEM to see the surface texture and to form the bioplastic films to see the texture. And then the biodegradation test was done in seawater and garden soil. And over here, we made a blend with PHB with PVA composite film. I'll explain the results further. As PHB has limited conditions as it is brittle in nature, so we made a blend with PVA composite to see the heterogeneous structure of it. So these are the results of the gram staining, which means that it is gram positive. In the primary screening, you can see the presence of black granules on each plate, producing that these strains have PHB production. And the microscopic screening was also done. You can see the black granules proving that the strains do produce PHB production. Our PHB extraction, out of the 3 strains, UE10 produced the most PHB yield, that is 43.9%. After that, when PBD was done in triplicates, and out of the 12 run orders, run order number 1 was selected, which produced the most PHB yield, that is 19.5 mg per gram. As you can see, the 3 variables, temperature, pH, and inoculum size, are significant, and Pareto chart visualizes the results showing that our factor single-handedly had a significant result on the production of PHB. CCD then again was also done in triplicates, which generated 16 runs on different temperature, pH, and inoculum size, keeping PBD factors same as well. And out of these 16 runs, run number 9 produced the most yield. That was 50 degree at 7 pH and 6.25 inoculum size. And the result was 90 PHB mg per gram. So over here, quadratic effect was seen between the interactions between 2 or more variables, temperature, pH, inoculum size, which showed significant results. And in Pareto chart, it visualized the results that the interaction between these 2 factors were significant. So in our contour plot, as you can see, the darker region shows the optimal range between the 2 interactions of pH and temperature on our substrate, that is PHB. That optimal range meaning that between 6.5 to 7.5 pH and temperature 47 to 52, you can see the optimal range of PHB growth. And same as with inoculum size and temperature, same as with inoculum size and pH. In our RSM, it was evaluated that at 47 degrees C, it produced 96.6 mg per gram of PHB yield. And after this, we got to know that out of all the screening, at this temperature, this specific pH, inoculum size, incubation time, and carbon source, it was evaluated that this temperature grows the most PHB yield. So these are the SEM characterization between the PHB film and then PHB containing PVA composite film, and then PHB free PVA composite film. As you can see in PHB, it shows a smooth surface of plastic. However, in PHB containing PVA composite film, it showed that at different magnifications, it shows the heterogeneous blend of all the film with the PHB. And in PHB free PVA composite film, it shows irregular surface, as in like it did not, that PHB did blend properly with the PVA composite film.
You have 1 minute to wrap up, please.
Then we did a biodegradation test in seawater with PHB plus PVA composite film dissolved in 30 minutes as compared to PHB-free composite film. And this was done in garden soil. However, these results are still awaiting. In 5 to 6 weeks, the results will be obtained. So as you can see, after the successful screening, we got to know that PHB is produced between the 3 strains used, and our statistical analysis showed significant results with PBD, CCD, and RSM. And in our biodegradation test in seawater, it showed that UE10 dissolved within 30 minutes. So these are my research contributes to the SDG goals. And good health and well-being, industry, innovation and infrastructure, responsible consumption and production, sustainable cities and communities, climate change, life below water, and life on land. I would like to thank my supervisor, Dr. 𝗬𝗓𝗍𝗂 Ijaz, for her continued support and guidance, and my technical lab staff that provided help during this stage, and my colleagues and my mentors for their continuous guidance as well. These are the references. Thank you.
Our next speaker is Ms. 𝗬𝗓𝗍𝗂 𝗍𝗏. Her title is Exploring the Relationship Between Genetic Variations and Lifestyle Factors and Obesity Related.
Assalamualaikum. Hello, everyone. This is 𝗬𝗓𝗍𝗏 𝗍𝗏, and today I'm here to present my research work that is entitled as exploring the relationship between genetic variations and lifestyle factors on obesity-related anthropometric and metabolic indices. So let's begin with the introduction. What is obesity? So the chronic imbalance in energy homeostasis leads to excessive deposition of body fat that ultimately results in obesity. Over the recent years, the prevalence of obesity has increased globally, and WHO declared that obesity as a global epidemic, and they reported that over 890 million adult individuals were obese in 2022, while 2.5 billion people were overweight. If we talk about our country, so Pakistan listed 9th among 188 countries in terms of obesity, according to the study conducted by Lancet. Obesity leads to various health impairment and cause disturbances almost all the biochemical processes in the body. So consequently, it increases the risk towards several comorbidities that includes stroke, dyslipidemia, cardiovascular diseases, metabolic syndrome, and certain types of cancer. So the etiology of obesity is very complex and multifactorial. There are several genetic, environmental, or behavioral factors which are involved in its manifestation. So why we are conducting this research, as I have mentioned in this particular slide, that each population has a specific genetic architecture and environment which offers an opportunity to conduct population-specific association as well as interaction studies. So the genetic association studies, they are very crucial because they will help us to identify potential genomic regions or the targets and its associated relationship with a particular disease. As well as the interaction studies, they are also very important because they will provide us valuable insight about the pathomechanism of a particular disease as well as its molecular basis, that what is the molecular basis and the mechanism behind a particular disease, complex diseases like obesity. So the objective of my study was to genotype the genetic variant that is FTO-RS9939609 and the 𝗬𝗵𝗏 RS7069102 in a sample of Pakistani population to find out their correlationship with the risks of obesity in our sample population, and lastly, to find out the interactive effect, to explore the interactive effects of these two genetic variants with the behavioral factors on obesity-related anthropometric and metabolic indices in our study population. So the outcome of my study will going to address the sustainable developmental goal, specifically target 3.4 that is related to the burden of the non-communicable diseases. It's not working. It's not working. So here comes the methodology. The study design was cross-sectional analytical case control study design, and the total number of samples was 612 participants containing equal number of cases and control. And this study was conducted at Dr. Panjwani Center, ICCBH University of Karachi, after taking approval from the Institutional Ethical Committee. So in this slide, I have mentioned about the method of sampling that I used in my research work. That is simple random sampling without replacement technique, and I recruited the study participant from the general population of Karachi after taking their written informed consent. So on the basis of the inclusion and exclusion criteria, we conducted the anthropometric measurements following standard protocols. And by using a validated questionnaire, we collected their information about their behavioral parameters. Along with that, we collected their fasting blood sample and proceed towards the downstream procedures or the experiments that includes the DNA isolation for the genotyping, and genotyping of both the genetic variants was done by using Tagman allelic discrimination assay via real-time PCR equipment, particularly Contest Studio 5, and the serum isolation was done to perform the biochemical assay in order to evaluate all these metabolic markers in our study population. So the collected data was proceed towards a statistical analysis by using SPSS software, and the genotypic frequencies were presented in the counts and percentage, and they confirmed from the 𝗬𝗏 equilibrium. Along with that, by using the additive genetic model, we sought or checked the association of these two genetic variants with the risk of obesity. And lastly, the interaction test or the interaction of these two genetic variants were also explored by using the linear regression, and all the statistical tests which show the p-value less than 0.05 that were considered as statistically significant results. So here come the results. Firstly, I have mentioned in this slide that represents the ethnic diversity that my study population showed that, as you can see, that the maximum population was from the Urdu-speaking background. In this bar graph, I have mentioned the genotypic frequencies of both the genetic variants that in cases as well as in control group. So both the genetic variants was checked or confirmed from the 𝗬𝗏 equilibrium. So as you can see in the slide. And the FTRS9939609, this genetic variant was checked for the association with the risk of obesity. Along with that, the 𝗬𝗏17069 and 102 variant also checked for its genetic association with the risk of obesity. Moreover, there was a significant interactive effect that was observed with the genetic variant FTRS9939609 with the aberrant behavioral factor, that is low physical activity, as you can see in the slide, that when these two genetic variants, they significantly interact with each other in order to increase all the obesity-related anthropometric phenotypes, including BMI, waist circumferences, hip circumferences, percent body fat, weight, height, waist to hip ratio, and skin pore thicknesses from 6 different regions of the body. Moreover, there was also a significant interactive effect that was observed between the genetic variants 𝗬𝗏1 RS7069102 with the behavioral factor. One is the irregular sleep-wake pattern, and the second is the low physical activity. When these two behavioral factors interact with this particular genetic variant, they increase the obesity-related anthropometric phenotypes in our study population. And lastly, there was also a significant interactive effect was observed between the 𝗬𝗏1 and genetic variant and behavioral factor, that is particularly low physical activity, to increase the risk of obesity-related metabolic phenotype, including the visceral adiposity index, lipid accumulation product, HOMA-IR, triglyceride, and fasting insulin levels in our study population. So in conclusion, we can say that there was a significant, as I have mentioned in my introduction, that obesity is a complex disease. So there are it is multifactorial disease. So there are multiple factors which are involved in its manifestation. So here in my conclusion, I would like to say that when these two genetic target, FTO-RS9939609 and 𝗬𝗏1 R7069102, when these two genetic variants interact with the aberrant behavioral factor, that is low physical activity and irregular sleep-wake cycle, they both interact with each other. They increase the risks or the predisposition towards the obese or overweight-related phenotypes in our study population. Lastly, I would like to acknowledge my research institute for providing me the funding to do this research work, my research supervisor, Dr. Sobia Rana, for her continuous guidance and support, and Jamil Rahman Genome Center for allowing me to do my genotyping work there. Professor Dr. Asma Salim for allowing me to use the gel doc system in her laboratory. My lab co-worker, Mr. Adil and Ms. Narjis Fatma, for their continuous support and guidance, and all the study participants who were included in this study. Here are the references of my research work that I have published from this work. Thank you.
Thank you very much for a very elaborative and presentation. So our next presentation is, it is an online presentation by Mr. 𝗬𝗏 Khan from International Islamic University, in vivo comparison of anti-snake venom activity of the honey-derived phenolics and polyvalent antivenom. Mr. 𝗬𝗏 Khan, online presentation. Mr. Aswat Khan, are you online?
Can you hear us? Hello, Aswat. Aswat Khan, can you hear us?
Yes, sir.
All right. So your audio link is working. We can hear you and we can see your presentation. Please start your presentation.
So, okay, sir. Are my slides visible to all of you?
Yes, your slides are visible to us.
Okay, sir. So, thank you for providing me such an opportunity to present my research work. So, let's get started. This study is all about in vivo comparison of snake antivenom activity of honey-dry phenolics and polyvenom antivenom. And I'm from Biotechnology Department of International Islamic University, Islamabad. So these are the contents that include background, introduction, objectives, and methodological representation, results, conclusion, and references. So let's get into the background. Snake bite envenoming is one of the neglected tropical diseases that has been declared by World Health Organization, and it leads to about 5 million people die from that bite bitten that occurred in mostly in Africa, Asia, and
Excuse me, Aswat Khan, please, if you could show the slides full screen. Slideshow. Yes, it is visible, but please put it on the slideshow. Excuse me, 𝗧𝗵𝗲𝗣𝗏𝗀, we are unable to see the slides. There's just one slide it is mentioned, slide number 4. It is stuck there. If you could refresh and then move forward. It is visible, but we can only see the slide 4, introduction slide. It's stuck there probably for last 5 minutes. Please change the slide if you could. We can see the cursor moving, but I mean, the slide is not being changed. Could you please close it once and then reopen it? Maybe it will help. Sure. No, it isn't. No, we cannot see the presentation. Screen share. Please go to screen share. Perfect. It is working now. Thank you. जी असफंद खान, क्या आप हमें सुन पा रहे हैं? आपकी स्लाइड शेयर नहीं हो रही है, अब दोबारा स्टॉप हो गई है. जी अपना माइक अनम्यूट कीजिएगा प्लीज़. आप प्रेजेंटेशन ओपन कर रहे हैं, अब आपने माइक ऑन म्यूट कर दिया है, अब अपना प्रेजेंटेशन ओपन कर लें प्लीज़. आप प्लीज़ अपना माइक अनम्यूट कर लीजिए अस्वथ खान. अस्वद हम आपको रिकनेक्ट कर लेंगे. आप वर्कआउट कर लें थोड़ा सा कि कैसे आप अनम्यूट होंगे और साथ प्रेजेंटेशन को नेविगेट करेंगे. मीनवाइल वील टेक वन प्रेजेंटेशन ऑन कैंपस यहाँ पे फिजिकली, और उसके बाद फिर हम आपको रीजॉइन करवा लेंगे. ठीक है जी. इनको कर दें वो भी ऑन.
a different hormone with different other combinations. So the best result we have got these multiple shoots with the MS medium with the 2 mg per liter of the thiazolone cytokinin with the combination of some other cytokinin keratin have got a very good result as compared to the others. These are the results of the in vitro shoot elongation and the root. Only on the simple media, MS media containing the 1 mg per liter of the IBA. So all cultures have shown the very good height in terms of shoot length, and also the maximum number of roots have been achieved. These are the pictures of the in vitro grown callus. 21 day, 28 day, 2 different media have got the very good result. Containing MS medium containing 0.1 mg per liter of the auxin Na alone, or with the combination of the other cytokinin 0.5 mg per liter of the BAP, and also same response have been achieved with the other cytokinin such as thiazolone. These are the results of the indirect organogenesis of this cannabis sativa plant. Grown plant leaves. So the in vitro plant growth yield of the protein, carbohydrate and lipids, so the higher yield of the in vitro grown leaves as compared to the native plant. So these are the results of the biochemical analysis done by the after the tissue culture, successful tissue culture of this plant. So these are the results of the green synthesized silver nanoparticle from the cannabis sativa in vitro enhanced cultures. The callus cultures have been used for the synthesis of the silver nanoparticle. So after the synthesis, as I mentioned in the methodology section, the method how we can synthesize that green synthesized nanoparticle, we confirm by the spectroscopic analysis the range of the 400 to 400 nanometer range, confirm the presence of the synthesized nanoparticle. Then we confirm the size of the nanoparticle by the scanning electron microscopy. The size of the nanoparticle around the 19 to 20 nanometer in size, and the elementary composition of the synthesized nanoparticle has also been performed. The presence of the quantity of the synthesized nanoparticle and other salts present in that sample. And also confirm the functional group by the FTIR in the synthesized nanoparticle, and also the non-samples means the normal plant extract. So these are the results of some biological activities such as anti-inflammatory, antioxidant, antimicrobial and the hemocompatibility assays. So in the anti-inflammatory assays, you can see that the silver nanoparticle, synthesized nanoparticle showing the low HRBC, low hemolysis, means the high protection for the or high anti-inflammatory potential, and also the antioxidant activity is concerned. So the good antioxidant activity also achieved in the synthesized nanoparticle, and also the synthesized nanoparticle gave the good result, and the low or the less hemocompatibility, which means the less than 0.5% of the hemolysis shown in the blood. So it means it is also acceptable when used as a drug. These are the antimicrobial results, but not that the any good result. So these were the last result of the fluorographic analysis for the estimation of the cannabinoids, specifically cannabidiol in our plant sample. First, we confirm on the basis of the colors, different samples, tissue culture, callus, shoot shape, in vivo and in vitro cultures, confirm the presence of the different cannabinoids in the range of the cannabinoids. Then we confirm the presence of the cannabidiol with the standard. Means the presence of the cannabidiol in our sample. These are the colors, color samples, presence of the color show indication of the cannabidiol, and after that we perform the HPLC analysis. It means that this, on the basis of retention time and UV range, the presence of the cannabidiol also shown in our in vitro cultures. So this is the conclusion of our study. In vitro propagation of the cannabis sativa, both direct and indirect methods were successfully optimized. Also, the biochemical estimation of the in vitro culture showing the good yield of the in vitro grown cultures as compared to the native or in vivo plants. Effective biomass production of the in vitro cultures were facilitated by the different elicitation methods such as methyl jasmonate, salicylic acid, and the synthesized nanoparticles. Silver nanoparticles that derived from the elicited methyl jasmonate treated cultures were successfully synthesized And then biological assessment revealed that the synthesized silver nanoparticle possesses a notable anti-inflammatory potential, as far as it has also shown the good antioxidant and the hemocompatibility potential. These are some grants that I have awarded from the Dai University of Health Sciences on this plant specifically, and the one last grant also secured by my MPhil student, which is this year, and the two consecutive years I have also successfully awarded this grant, which is very difficult to achieve in Dai University of Health Sciences. So 2 years grant, so alhamdulillah I got these two and complete the first one, the second one. The first one is already completed, and the second one, which is the animal model study of the synthesized silver nanoparticles, will be performed inshallah in this year. So in the last, I would like to acknowledge the Vice Chancellor's in-fund initiative of the Dai University of Health Sciences of providing me the facility to perform this work, and also my graduate and undergraduate students, 𝗟𝗏𝗄𝗏, 𝗬𝗏, 𝗟𝗏𝗙𝗏, 𝗧𝗵𝗲, and 𝗪𝗏, who performed this work, and this work made successful. So thank you all for your patience.
Okay, so it's already a tiring and a long session, but we still have two presentations. I would like to 1.5 presentation actually. So Mr. Aswat Khan's half presentation is remaining. So he will continue from the same point. I think he just discussed the methodology. So Mr. Aswat Khan.
Are you here? Hello?
Yes, ma'am.
Okay, so then please share your screen for presentation.
Okay, ma'am.
And please start it from the result section.
Ma'am, is it visible to you?
Yeah, it is visible.
Should we start from methodology or
No.
results?
From results. Yeah,
Yes.
that's fine. Thank you.
Ma'am, this was the in vivo study of the 𝗏𝗏𝗍𝗏 extract and venom, and there were different groups that include control group, experimental group. So yeah, each group had 3 𝗧𝗵𝗲 and 𝗵𝗏𝗍𝗏, and the control was included in which the venom was injected in the mice, and it was about 69 microgram, and another control had phenolic extract, that is phenolic compound that was able to neutralize the venom toxins in the mice. And third one is that the control is also anti-venom that was administered in these mice, and this was about 110 microliter. And fourth one is venom plus extract, and this was given in a group like in intravenous. Another one is venom plus extract that was given through interperitoneal route, and sixth one was the combo, the venom plus extract but anti-venom, and last one the honey, that was a crude honey plus venom plus, and the route was interperitoneal. So in the control group, all the mice, all the model animals that mice die. Another group control extract, they were the survival rate was 100%. Another control group to whom the anti-venom was administered was also survival rate 100%. Then was phenolic extract plus venom. Then we went to the partial characterization of venom. This was conducted through the STS page, and we got the three major bands that corresponds to the venom toxins that are majorly responsible for different tissue damage and tissue necrosis and other amputations related to snake bites. Then we went to the characterization of antivenom, and we checked the percentage of phenol that is that act as a preservative and is necessary for the stability of therapeutic antivenom. This is the physiochemical characterization of honey, and we had two sample honey, Kashmir honey, and other was Saudi honey, and basically the Saudi honey had more reducing sugar as compared to Kashmir. There was acidity less than 1%, both of them, so it means they had good antimicrobial resistance, and so there was more or less than 20% of moisture, and the standard one is 20 or less than 20, and reducing sugar content was higher in Saudi honey as compared to Kashmir honey. Then is the microscopic analysis of honey, and it is, you can see there was no bacterial or yeast growth, and you can see the bubble formation or wax type structures, and it was also confirmed through the microscopic analysis that their botanical sources was majorly from trees. So this is the quantitative analysis of phenolics compound, and it was done through the spectrophotometry, and we had standard gallic acid. So you can see the results that majorly the gallic acid was present as a active bioactive compound in both of these samples, and they both have ability to neutralize the proteins, enzyme proteins that is present in the snake venom, that are majorly metalloproteases and three-finger toxins and phospholipases too. And conclusion, both honey has samples. They successfully neutralize, and the survival rate was 100% of these phenolic extract. And once successful, it can be there can be clinical trials of these extract, and these phenolic extract can be given as a oral product or as an injectable drug, and they can counter the problems that are related to the affordability and reactions that are related to the conventional antivenom administration, and these are quite natural. So yeah, it will induce less allergic reactions in the patient to whom they are administered. So yeah, this is the conclusion. So future prospects include to develop some non-conventional therapeutic drugs to increase the efficacy of antivenom products, or they can act they can complement the efficiency of available antivenom products, and open the doors to the new therapeutic drugs. These are other references. So
Thank you so much, Aswat Khan, for the presentation and the patience you had. Indeed, it was nice, and sorry for any inconvenience in between. So our next and even the last speaker of the day, take a this is Mr. Mohammad Mansoor Majid from the SZABIST University, and his title is Association of the IL-10 single nucleotide polymorphism and its correlation with the IL-10 cytokine levels in chronic periodontitis patients and healthy individuals. You may start with the introduction yourself.
Okay, Assalamu alaikum. This is Dr. 𝗬𝗵𝗲 and I am a PhD scholar in SZABIST. I will not take much time. My presentation. Okay, my presentation is about genetic polymorphism of IL-10 gene in periodontitis in Pakistani population. So its objective is to check the association of IL-10 polymorphism and its salivary cytokine level and its correlation with the clinical disease. So first of all, periodontal disease are the disease that results in the destruction of tooth supporting structure, and because of this, we may observe the mobility in the teeth. So in this disease, they have multiple types, and we chose chronic periodontitis. Why this disease? It is a highly prevalent disease. If we talk about the Pakistani population, around 60% we observe cases of mild periodontitis. It has multiple types, mild, moderate, and severe, and really 20% of moderate, severe cases, which results in tooth extraction. Multiple factors are associated with this disease, and one of them is the single nucleotide polymorphism in certain gene. So this study is to find out the association of IL-10 gene at -819 locals and its association with its salivary cytokine, and results, it's with the clinical correlation. So according to the literature, single nucleotide polymorphism results in too many activities in which the pro-inflammatory genes, which are like IL-10, IL-1β, and anti-inflammatory gene IL-10 are responsible for periodontitis. Multiple studies have reported their significance in this, but unfortunately, before that in Pakistan, no studies were done on especially this disease. I'm talking about periodontitis. So this disease has, that's why we have conducted this study. And as you know, there are multiple genes which are involved. Most commonly is IL-1β, which is a pro-inflammatory gene, and IL-10, which is an anti-inflammatory gene. And in near population, like in India and Iran, we have observed that their role is very significant in periodontitis. So why I have chosen IL-10 gene? It is an anti-inflammatory gene, and its cytokine, which is IL-10 cytokine, if its production is reduced due to polymorphism, the role of pro-inflammatory gene increases. For example, IL-10, as we know, that it stops the production of excess production of IL-1β and TNF-alpha. So in other case, the production of those cytokines increases and results in the destruction of the bone and other periodontal tissues. These are the diseases which are also associated with periodontitis, and they have also linked with the genetic polymorphism, single nucleotide polymorphism. So it was a cross-sectional study conducted in Dow University. Initially, we checked 3,000 individuals. Later on, the sample of 122 patient individuals, in which 59 were control and 62 were patients were analyzed as they had a good or better PCR results. And later on, genotyping was conducted from macrogen, Sanger sequencing, I mean. We had certain inclusion and exclusion criteria. This was the instrument that was used to check the clinically periodontal, to perform the clinical examination for periodontitis. We collected the blood from some patients, did the DNA extraction, performed PCR, then gel electrophoresis, and sent to macrogen for sequencing. These are the images. Alignment according to Omega software. These are the images which we obtained after the sequencing. And for saliva, for cytokine level, we did saliva collection and centrifuge it, stored it, performed ELISA, then analyzed and checked those things. So we basically checked these outcomes, which were genotyping, single nucleotide polymorphism, and mainly the clinical outcome of those. So initially, we didn't find any significant difference in the gender, male, and these are features which may be related with the disease modification. So we didn't find any, but definitely in patients and in healthy individuals, there was a significant difference in clinical parameters. These were the percentage of genes. So here we can see the distribution of alleles. In healthy individuals, majority were with the T alleles, and in diseased, we had C allele. Similarly, if we can see the electropherograms, later on the genotype, CC genotype, was more common in and significantly higher in a patient as compared to the TT genotype, which is significantly higher in healthy individuals. And this is the criteria that we observe mild, moderate, and severe cases, that in mild cases, majority of the patient, majority of the individual were with CC genotype. And in severe, it was with TT genotype. So, about the cytokine level, significantly, if we check, the cytokine level is significantly lower, IL-10 cytokine level is significantly lower in periodontitis patient. So it means the anti-inflammatory cytokine is lower in this disease. So that's why it is causing this disease. We can check here. And similarly, it is low in severity of the disease. As the disease severity increases, the level of cytokine of IL-10 decreases. Clinically, we observe, and it is positively correlated. Now the discussion. Mainly IL-10 is the cytokine which is linked with the stoppage of certain pro-inflammatory cytokine. Moreover, if its level decreases, the activity, because there is anti-inflammatory, so inflammation increases. For example, the destruction of bone, periodontal ligament, even it destroys the cementum of the tooth. And multiple studies have shown the similar result. Few studies in Caucasian population have some different result, but studies in India and Iran have the similar result, and our studies are also in relation with these studies. So we found that single nucleotide polymorphism of IL-1 is responsible for or is associated with periodontitis in our population. And there are certain limitations. Number one is the limited sample size due to limited budget, and some strength that it is among the initial studies conducted in Pakistan. And more genes, for recommendation, more genes should be studied with larger sample size, and this study was conducted basically in Karachi. So if we conduct a study throughout Pakistan or particular population, we may have some different result. Okay, thank you so much.
Okay, this is the end of the presentations. So if you have any of the questions from any of the presentations, if the presenter would be here, then definitely he would be answering your questions. So any questions from the audience?
I have a question to Mr. Your presentation was very good. It was about AI and the effects of AI, how this AI is affecting us, and how is this AI can be, or the phenomena of AI can be beneficial to us. So while you were doing this systematic survey, what were your findings, that what are the jobs and occupations that will be totally wiped out by the AI, and what are the jobs and occupations that will be have zero chance or negligible chance to being affected by whatever the AI can achieve? They cannot wipe that out, or they cannot affect that occupation.
Thank you, sir. Sir, since I mentioned that my work was based on more of a aspect that the AI and human, and AI, the perspective of AI and perspective of human, both their inputs are valuable, and neither I thought nor I worked that what kind of job will be eliminated. I worked on the positive sides, how can they, both of them can augment. there are theories, there are, or we can say conspiracy theories, or there are suggested theories that this job will be wiped out, or this job will. But the focus of my study was how we can augment, how we can coexist in coming future, in a better future, how we can coexist in every way possible and in all the fields in which AI is finding its use cases. How we can incorporate the input of humans with the input of AI. I did not, unfortunately, I did not include what kind of jobs are going to be wiped out, or in a severe sense, or in a low sense, or in a moderate sense. Hope I answered the question.
Yes, you have definitely. But actually, I was worried about I was more worried about my occupation. We are mostly teachers here. In fact, in the first 2 years, then we have students. So I was thinking कि मेरी job तो नहीं कहीं खतरे में. So yes, so that is one of the things that I wanted to know. Thank you.
Sir, I have a question for Dr. 𝗬𝗓𝗏, who is my graduation senior and my dear friend. I am always a fan of his work. My question is that he mentioned apoptosis. Cardinoloids are targeting cancer cells through process of apoptosis. So what were the methods for screening that you applied that you became so sure that they are going to only target the cancer cells in terms of apoptosis and not they are going to target other cells? So this is my question.
Thank you, Taha, for asking the question. Yes, the cardinoloids, because so many different genes which include the BAX, caspase-3, caspase-9, some executioner caspases. Other than this, I also checked the different pathways like 𝗬𝗵𝗃, and because 𝗬𝗵𝗃 is the transcriptor, major transcriptor in the cancer. Perhaps from this, I also checked some different pro and anti-inflammatory, which includes the 𝗧𝗵𝗲. This 𝗧𝗵𝗲 gene is pretty much interesting in our case because the BCL2 in my case, I never got the positive result, but once I checked the 𝗵𝗃5, that is the 𝗧𝗵𝗲 gene, it reduces down in the after the usage of the cardinoloids. BEX was also uplifted. Caspase was also showed the greater gene expression. RAS pathways was also reduced down. Beside from this, I also checked the AFM results along with this, the same. So these showing that the apoptotic bodies were there. The overall cell architecture was also disturbed. Cellular changes along with the cellular changes and the pathways along with the protein. I also checked the immunocytochemistry, which include different kind of proteins, in which I have checked HIF-alpha, that is the hypoxia-induced factor 1. Apart from this, I also checked the BEX, KRAS and HRAS. So on the basis of these different parameters, which include the gene parameters, proteins, AFM study, yes, how can I forget the mitochondrial pathway? Because mitochondrial pathway is very much interesting in case of a cancer, because the production of the cytochrome C was also decreased, and this is the main reason why cancer cells get evaded from the apoptotic pathway. So overall, in a nutshell, because the whole work was done in ICCBS under the supervision of Professor Dr. Shabana Simji. So along, in a nutshell, I conclude that using different pathways, I am conclusive that cardinoloids may possess a strong apoptotic and anti-proliferative action.
Thank you so much. I would request the students to be calm, because this is the end of the day. I want see the patience from yourself. You have been talking, you have been sleeping, I know. So please, This is the last session of the day. Any questions? The intensity of your voice let us know that you are almost, but not almost, you are tired. Drained, yes. So if you don't have any questions, so let us conclude the session. Yes.
The scholar that gave a presentation on melanophyllin. I have a question that your presentation was remarkable and your effort was also very remarkable. We must appreciate his effort of him coming here like this. My question is that the technology framework that you discuss, single nucleotide polymorphism, can this technology framework be used in other traits of human genomics other than coloration? Can this be used?
Actually, single nucleotide polymorphisms are of different type in different genes. But the gene of interest on which we have worked on is basically on the coloration activity. Otherwise, we can also target different genes which are responsible in cancers also and different diseases also, in which we can find other single nucleotide polymorphism, and we have a broad ground that we can work on these single nucleotide polymorphisms. But basically, my team's own interest was on basically rabbits, and due to and I have mentioned earlier that due to genetic similarity, and there is basically no working done on the Pakistani local breeds of rabbits. So that's why it gave us a novel opportunity of a study to make work on them. And as the coloration activity is occurring on the fur of a rabbit, there is the same process of similar protein in humans also, which is giving us coloration activity. As you may see, Africans, those have darker skin colors. As you may see, some English people who have very lighter skin color. So basically, this is a gene which is responsible for giving this kind of color to the human skin. So basically, my target was on the coloration activity, but there can be several genes which are responsible for the different kinds of diseases and other things also, in which we can identify single nucleotide polymorphisms. Thank you.
So it's time to conclude the session. It was really nice to be here, right from lunch till now. So the session was started at 2 p.m. With the title investigation of the role of the novel cardinaloids by the Dr. 𝗍𝗫𝗄𝗧. Then we taken different presentations in between, like in vivo comparison of the anti-snake venom. The presenters were students, faculty, professors. So you see there is a range of the presenters we saw in between, and this was wonderful that this consortium of the scientists, budding scientists till the experienced scientists, we really enjoyed, and hope this would be really great for the student as well. And definitely these kind of the conferences, seminars, and the other like presentations are really good for the students and are really arranged particularly for the students, so they can be benefited. And I hope you would have been taking a lot of advantages out of these presentations. So and from us, I am thankful to the organizing team, particularly the Dr. Rahman, Dr. Kashif, Ms. Urusa, Dr. Urusa, and other team as well. Thank you so much from my side. And I would request also Dr. Zulfikar to have some words with us.
take much time. I just want to say that this is a very good forum to learn, and we saw that there are only 2 or 3 questions, but I would suggest that you get around and talk to the people and try to learn from them. And this is a very good effort made by SZABIST, and they are normally organizing these conferences. Thank you very much.
Okay, so with this, now I am the moderator of the concluding session, and I would request Dr. 𝗬𝗓𝗏 and Dr. 𝗧𝗵 to please come on the stage and commence the certification distribution. Thank you. Okay, I would to please join us on the stage. So, may I please have Dr. 𝗂𝗏𝗄𝗏 on stage to receive his certificate of appreciation. We are also giving hampers to our oral presenter, which are sponsored by different industries, so, and we are very thankful for that. May I now have Ms. 𝗬𝗏𝗄𝗏 Shahid on stage to receive his certificate? Okay, so may I now have Ms. Hina Nawaz? May I now please have Mr. Mohammad Faraz on stage to receive his certificate? Now please put your hands together for Mr. Taha Ahmed. May I now please have Dr. Shiba Naz to come on stage to receive her certificate. Okay, so in a round of applause, please welcome Dr. Mansoor. And now please welcome Ms. Dania. May I now please have Ms. Saja to receive her certificate? Okay, last but not the least, I would call Mr. Adil to receive his certificate. Now I would request Dr. Professor 𝗵𝗲 𝗂𝗏 to give shield and the souvenir to our respected session chairs, Dr. 𝗧𝗵𝗲. Now I would request Dr. 𝗵𝗲 to present a shield to Dr. 𝗬𝗃. So I thank you all for being a part of today's session, and with this I am signing off. Have a lovely evening and see you tomorrow. Allah Hafiz.